Good morning, everybody. I am attempting to make a video for you guys just to go through the important points that I want you to know for our Unit 5 test that is coming up next week. So let's see, we're talking about reproduction and development. Reproduction and development are necessary for the continuation of any species. So that's the reason why any species reproduces, because they want their particular genetic traits to be passed on. They want their species to, not become extinct. So the reason that reproduction happens is for species to continue, and all species have unique related strategies for reproduction. We talked about a lot of different strategies this unit: chickens, brine shrimp, salmon, humans. What else did we talk about? I can't think of anything else. But anyway, so we talked about a bunch of those, and reproduction is a life process which living things produce other living things of the same species. We can't produce things, offspring that are a different species than the parent. That's kind of obvious. I'm sure you guys knew that already. So focus on things that have the stars on them. That doesn't mean you should just skip through everything that doesn't have a star, but focus on the things that have this little star in the corner here. We know that there are two types of reproduction of organisms. One is the sexual reproduction, which we'll talk about in a little bit, and then there's asexual reproduction. What you need to know about asexual reproduction is that it is from one parent. So there's only one cell or one organism that divides into two or more identical organisms. So it's a single parent, one cell, or one parent, depending on what type of organism we're talking about, producing offspring that are genetically identical. There is no variation in the DNA. So I think this is kind of funny. What if humans reproduced asexually? This was a human. He would just split in half. This would be like mitosis, split in half and have two humans that are exactly the same. That just doesn't happen. And I'm kind of glad it doesn't, because it's kind of frightening. All right. And what if this is one called budding. So this would be like mitosis or fission. And this one would be similar to budding, where you have this tiny little sprout growing out of the side of an organism. It buds off and develops into a full-grown adult organism. So I'm also happy that that doesn't happen in humans, because that's also, like, especially this part right here, that's a little scary. Okay, last but not least, asexual reproduction. This would be like regeneration, for example, of a starfish. There's also a class of animals called cnidarians that do this all the time. We have some of those little anemones in our classroom. This would be if a piece of the organism, say a plant or the things I just mentioned, fell off and developed into an entirely new organism. So asexual reproduction, organisms are genetically identical and there's only one parent. Here's some examples. Binary fission of an amoeba. You have a single-celled organism right here, duplicates its chromosomes and then splits in half, and you have two organisms that result. And these are genetically identical, and there's only one parent. Same goes for this lovely little paramecia here. You start with a single parent. It duplicates all of its stuff, and then it splits in half, and you have two identical paramecia that are genetically identical to the single parent. You can see they have some variations in the way they look, but genetically they are absolutely the same. There's no genetic variation at all. This happens in some plants. You might see this in your garden or in your house. They will send off these little runners, a single plant. There's no flowers here that are accepting pollen from another plant. We know some plants reproduce sexually, but not this guy. And he sends off a little runner, and an identical plant, genetically identical plant. You see it has fewer leaves and it's a little smaller right now, but it's genetically identical. That's going to sprout up. Same for yeast. These guys bud. So they put a little bud that kind of splits off from the mother cell, and these two are genetically identical. This one will eventually grow into a full-size yeast cell. Last but not least, this is a hydra that is in the cnidarian family. You can cut this guy in half. He's an aquatic creature. We have some that live in our bays nearby. You can even cut him down to this little tiny nub right here, and they'll grow into 2 full-grown hydras that are genetically identical, and there's only one parent. So these guys have the same DNA as this guy and this guy, exactly the same. Okay, sexual reproduction involves 2 parents. That's the first big difference. Whoops. The offspring are genetically different from the parents. So we have different DNA than mom and dad. Your DNA is very close. The DNA of the offspring is very close to the DNA of the mother and father, but it is genetically different. If you were genetically the same, you would look exactly like your mom or your dad. The sex cells are gametes. Those are the eggs in a woman or a sperm in a man. And these eggs and sperm are formed by the process of meiosis. That's the only time we see meiosis in our body is during the production of eggs and sperm. So any other cell division that we talk about, or differentiation, healing, disease, anything like that, they're all going to be mitosis, because meiosis only happens when you're forming your sex cells. The gamete and the sex cells will fuse during fertilization, and the fertilized egg is called the zygote, which you guys all know, and it divides by mitosis to form new cells, and then a new organism. Alrighty, what do we have here? So the basics that you need to know about the sperm and the egg. Look how huge that egg is compared to the sperm. The egg is actually, I think, the only human cell that is possible to be seen with the naked eye. So it's pretty big as far as cells go, but those little sperm are tiny. The eggs can't move, the sperm can. The eggs are produced in the ovaries. In fact, females are born with all of their eggs. So when this is happening, this is actually happening in females when they are still in their mother's womb. So you're born with all of your eggs. All of your eggs are made, if you're a woman, when you are still inside your mom's womb. Men have sperm that are produced in their testes, and they continue to make them throughout their life. Oh, I spelled throughout wrong. Don't look at that. Eggs are very large in size. Sperm are small. Eggs have some stored nutrition, so that as the embryo begins to develop, it has some nutrients to pull in before the placenta is formed. Sperm do not have that. The female reproductive system is regulated by estrogen and progesterone. Those are the hormones that regulate, that's an important word, they regulate the reproductive system, whereas in males, this is regulated by testosterone. Do not worry about Octomom. All right, male reproductive system. You just need to know that sperm are produced in the testicles. You have these lovely 2-sided front and view side here. This is where the sperm are produced. They're produced throughout the life of the man by the process of meiosis. And then this is where they're transported through the vas deferens. You should know that what would happen if this was blocked? Would fertilization be able to occur? Just know a little bit about be able to imagine what would happen if there were issues with the testicles, what would that affect, or what would happen if the sperm could not be transported. All right, now the female reproductive system, we've talked a lot about this. You can kind of forget about this side view. I saw it like once or twice in the exams, but really mostly we want to focus on the front view. So we know that we have the ovary where the eggs are produced, and this is where the eggs are stored from birth through the remainder of the woman's life. The fallopian tubes, we've got 2 of them. This is where ovulation occurs and fertilization occurs. Did I spell that wrong too? What the heck? Okay. And then uterus. I really do know how to spell fertilization, I promise. The uterus is where the embryo implants and grows. So after fertilization, the embryo travels down to the uterus, kind of finds a little cozy place in there, and that's where it will develop for the next 40 weeks. Okay, so the sperm meets the egg in the fallopian tube. This doesn't happen in the ovary, doesn't happen in the uterus. It happens in the fallopian tube. Each of these cells, we start with the egg and the sperm. The process of meiosis results in cells that have half the number of chromosomes as every other cell in your body. So humans, we have 46 chromosomes in every cell in our body. That's a full set. So our eggs and our sperm are only going to have half a set, or 23 chromosomes. And that's because when they meet and form a zygote, they're going to make a brand new set of 46 chromosomes. So there's that. Fertilization in humans, A mature egg is released once a month from the ovary. 1 month it goes from that ovary, the next month it goes from that ovary. And if it meets a sperm in the fallopian tube, it is fertilized. And the resulting cell is called a zygote. Now has a full set of genetic information. That's 46 chromosomes, half from the dad, half from the mom. So the new cells start to multiply, and the zygote travels down the tube, the fallopian tube, toward the uterus. It's dividing by mitosis. It starts as a single cell, and then it's 2 cells, then it's 4 cells, and it's 8 cells, until it gets to be about 100 cells. And then around 100 cells, this is called a blastocyst. It's actually like a hollow ball. There's a few layers of cells in here that are starting to form, and then there's a single layer of cells around this. It's about 100 cells, and at about 7 days after fertilization, it finds a little cozy place to stick into the uterus, and this is where it will remain. The uterus, the job of the uterus is to support the development of the embryo. That's where the embryo will grow into a fetus. This is not anatomically accurate. Babies are not born wearing diapers. But they implanted it. He's just so cute. I had to put him in there anyway. So the embryo stays here where it grows until it's ready to be born. So it's 40 weeks. And the cells continually divide and divide and divide by mitosis millions of times until they've made a baby with different shapes and functions. All the cells have different shapes and functions. The purpose of the placenta. The baby is nourished through the placenta. So the mom's blood is here-ish. And nutrients pass from the mother's blood into the placenta. The blood isn't mixed together. There's no mixing of the blood. It's just the nutrients diffuse across the membrane of the placenta, similar to the way that we saw oxygen and carbon dioxide diffusing through the alveoli of the lungs, or the way that we saw water diffusing through the cell layer of the onion. Those nutrients diffuse through the placenta. And then they go through the umbilical cord into the belly button of the baby, where he takes all the nutrients that he or she needs, goes through all kinds of metabolic processes because he's doing or she is doing a lot of growing. And then the waste is exported back through the umbilical cord, through the placenta, where it gets diffuses back into the bloodstream of the mom and is excreted with her waste. So the blood cells don't mix. The bloods don't mix. Although, well, just a little side note, researchers are finding fetal cells in the bloodstreams of moms up to like 10 or 15 years after they're born. So anyway, that's kind of cool. But the placenta can filter out some toxins, but it can't filter out all of them. So some harmful things, if the mother is exposed to pesticides or chemicals, they may pass through the placenta into the baby, causing difficulty with development. The amniotic sac, which is this shown by the white here, that's like a little clear balloon that this guy is living in, and it's full of fluid. And that helps protect him from bumps and jostles and stuff like that. All right, you don't need to know all the details of mitosis. You do need to know when it's used, and you do need to know that the DNA is replicated before mitosis happens. Otherwise, we'd eventually run out of DNA. So before mitosis happens, the DNA is always replicated completely. And you need to know that the daughter cells have the exact same DNA as the parent cell. But you don't need to know all these stages. I'd like you to know them because I think it's important, but you're not going to have to tell me them for the test. Meiosis, we know that's the process that is used to produce sperm and egg cells. What I would like you to know for this is basically that the DNA is replicated first before meiosis occurs, so that you have enough to go around. Additionally, you have to know that the daughter nuclei, so these would be your egg cells or your sperm cells, they're all genetically different than the starting cell. So look at this, if you start with chromosomes that look like this, and all of a sudden this one's got a little bit of variation. They each have some genetic variation, and this is where your genetic variation as an offspring comes into play. So each egg cell is genetically different, and each sperm cell is genetically different. And those differences also help contribute to evolution. Mitosis and disease. Just know that mitosis out of control can sometimes be contributing to cancer, where you have tumors, cells just keep replicating without being subject to their normal checkpoints. Mitosis is involved in wound healing. These are some basic concepts. If you lose some skin cells, mitosis is going to help bring those skin cells back in there. It's going to help replicate those skin cells. You should know this stuff. Okay, differences between mitosis and meiosis. These are the basics. I just made a little chart for you to have it all in one place. This you should know about reproductive strategies. Why do some species have a few offspring at a time, or one, like humans, or two? And why do some have thousands and thousands of babies at the same time? And you know, based on our brine shrimp, what was it, the reproductive success for our brine shrimp, even in the best salinity, was only like 5%. So you know that brine shrimp have to have thousands of babies in order to just get a handful that are going to survive. So it has to do with the rate of survival. This is just kind of a little side note. Why do chickens have to have internal fertilization? Their eggs are developing outside. It's because the shell, this is the egg, travels down this kind of disgusting looking track where it gets fertilized by the male. But the fertilization has to happen before the egg is laid, because the shell is put on the developing egg like right before it leaves the chicken's body. If it was already hatched, it would be too late for the male to fertilize it, because the sperm couldn't get through the chicken egg shell, because those things are hard. All right, here we go. Differentiation. All of our cells in the very beginning, when we're just that blastocyst that embeds in the uterine lining, are all identical. We all have the exact same DNA in every cell of your body. In fact, this stem cell has the same DNA as this nerve cell. Same DNA of each of these specialized cells in the retina. Same DNA as what's in the muscle cell, and same DNA as in the red blood cell. How on earth, if DNA is the instruction for what to do as a cell, how on earth are these looking so different if they have the same DNA? They do completely different things. And the answer is they become differentiated by using, where do we go? They have the same DNA, but they don't use all the same DNA. They use different instructions. So say the muscle cell is going to use this little piece here that says make actin or myosin. We haven't talked about those yet. All right, so let me choose a different one. Say this is a nerve cell, instruction for a nerve cell. It might use this gene right here that says make a synaptic vesicle protein, or use this little gene, and use this over here that makes an axon terminal, and use this one over here that makes a neurotransmitter receptor. Those would be the instructions that the nerve cell would use. It doesn't use all the DNA. It only uses the little parts it needs. So each cell of your entire genome each cell has the entire genome, but it uses different parts of the DNA. It uses only the genes it needs. Genes that are turned on are expressed, turned off are repressed. And what else? We have a little bit more. Okay, so know what each of these is, how you get from A to B, and what they're called. Fertilization, mitosis, differentiation, growth, things like that, right? So this is differentiation, where the cells start to specialize. This is mitosis, where you're just dividing. This is also mitosis too, by the way, but anyway. And know the order. Gametes produce a zygote, produces embryo, that produces a fetus. You already know what the amniotic sac and the placenta do. Okay, this is something we have not talked about yet. I'm going to bring up when I see you, but it's an important point. Can the health habits of the father and the mother affect the embryo even before fertilization? Think about that for a second. If you have all of your eggs in your ovaries, the things that you do, the toxins you're exposed to at any point, can affect those eggs. So smoking and other toxins can cause DNA mutations in the sperm as they develop in the testes, and in the eggs that are sitting in the mother's ovaries. So mutations can then be passed down to their offspring. So just like smoking can give you mutations in your lung cells that can cause lung cancer, it can also cause mutations in your eggs or your sperm that could possibly affect the health of future offspring. In fact, this study right here, it says paternal transmission, that means from the dad, of altered DNA may compromise embryonic development in utero, resulting in failed implantation, early pregnancy loss, or disturbances of postnatal, that means after birth, development. So in this case, they had 642 individuals of a Chinese community where female smoking is discouraged, but paternal smoking was not discouraged, apparently. So they found that paternal smoking during preconception, that's before there's any fertilization or anything, paternal smoking increased the risk of childhood cancer within the first 5 years. That's kind of scary, but it just goes to show you that before fertilization, the health of the mother and the father are both important, and avoiding toxins throughout your life is very important. Why are drugs and alcohol so much more dangerous for an embryo and a fetus? Because the fetus is smaller, so anything that is put into the mom's bloodstream is going to be really more diluted for her. She probably has something like 10 to 12 pints of blood, where the baby only has about a cup, which is really a half a pint. So anything that's in her bloodstream that filters through could become more concentrated in the fetus and cause a much more dramatic problem. And the organs of the fetus are still developing. So if you have a problem during development, the organ may never form properly. So for example, we looked at this quite a bit. If you're exposed in an embryo to a toxin that affects the heart at, say, 4 weeks after fertilization, there's a chance that heart may never form properly and the fetus could be born with heart problems. If you are exposing the fetus to something that's a known teratogen or toxin for development of the eyes, at around 7 or 8 weeks, you might never have proper eye development. If you expose them later, you're going to have a less chance of major problems with the eyes here. But the point of this is that toxins can get across the placenta membrane, and having toxins affecting the fetus in these early stages can prevent proper formation of organs. And therefore, the baby can be born with congenital, which means with birth, defects. All right, and last but not least, we look at a lot of embryos today, or this week, and we saw that at the early stages, let's look at stage 1, I think we had this guy, they are almost impossible to tell apart. We had the human, the calf, the chick, the fish. What else are we dealing with? Oh, a pig. They looked almost exactly alike at these early stages of development. And the reason for that is because we are all evolutionarily related. So all of those embryos have common structures because they're all evolved from the same life forms during early evolution. All organisms on life evolved from bacteria, and we've all evolved with similar structures that are adapted to live in specific habitats. So humans, we looked at the mammals, humans, calves, and pigs all had features that looked more identical at the later stages, and that's because we're more closely related evolutionarily. So you can look at the structures that an organism has. I think that's here. You can look at the structures that an organism have, like arms, legs, teeth, beak, anything like that, to see if they're genetically related, see if they're evolutionarily related. Like these guys both have thumbs, so we're more evolutionarily related. All of these guys have fur and mammary glands. Those are milk-producing glands, so we're more evolutionarily related. We can also look at DNA sequences. So we could sequence the DNA from this guy's genome, this guy's genome, this guy's genome, and we could compare them and see how similar they are. And you need to know that sexual reproduction, that means we saw before how the resulting cells from meiosis are genetically different from the parent cells. So that sort of variation in DNA and sexual reproduction leads to new combinations of genes, which helps lead to further evolution. All right, I think that's it. Let me know if you have any questions. Come prepared on Monday with questions that you may have about anything we've done. All right, I hope this works. Goodbye, everybody. Have a great rest of your weekend. I don't know how to stop this. Did I not even, what the heck?